Hello students, welcome you all to a new video of Sunanda's tutorial. So today in this video we will discuss about the detailed steps of transcription process in case of prokaryotes. So at first, what is transcription? So protein synthesis inside the cell takes place in two steps. The first step is transcription, where the DNA is copied to mRNA to form mRNA in the process of transcription. Then in translation, that mRNA or messenger RNA is decoded to form linear chain of protein or polypeptide chain. So it is the first step of protein synthesis. We will discuss the process of protein synthesis, the first step of protein synthesis in prokaryotes. So at first, what are the machinery, what are the tools which are required for transcription? Prokaryotes rely or depends on one multi-subunit enzymes, the RNA polymerase with multi-subunits, many subunits. The enzyme alternates between two functional states. So this enzyme have two functional states. One is core enzymes. Core enzymes consist of alpha, beta, beta dash and omega. This complex process possesses the catalytic activity required to synthesize RNA but cannot identify the promoter on its own. So for initiation we have to identify the promoter, but core enzymes cannot identify the promoter site. It requires hollow enzyme consist of alpha, beta, beta dash, omega, along with that sigma factor. Sigma factor along with core enzymes are required for identification of promoter. So hollow enzyme formed when a temporary specificity subunit, the sigma factor, binds with the core enzyme. The sigma factor directly recognizes the promoter sequence on the DNA. Okay. So these are different subunits of RNA polymerase which are required during transcription process. So now what are the steps? So the whole process of transcription is divided into three steps. Step 1, initiation, then elongation, then termination. So at first, initiation, the transcription initiation requires sequential steps to transition from a resting DNA molecule to actively extending RNA chain. Then promoter recognition, the hollow enzyme consisting of alpha, beta, beta dash and sigma factor, they recognize the promoter sequence. They scan the DNA and locks onto the promoter region. They binds to the promoter region. So in case of bacteria like E. coli and other prokaryotes, it identifies two highly conserved consensus sequences located upstream of the +1 transcription start site. So the upstream, it is located in the upstream of +1 transcription start site. What is upstream sequence? It refers to segment of DNA or RNA located before a specific point, usually before transcription start site. That is referred to as +1 site. Okay, and upstream. So it recognizes -35 element, typically sequence TTGACA, which serves as primary recognition point. Another one -10 element or Prinobox. Typically sequence TATA80, which is rich in adenine and thymine, making the strand easy to separate. Okay. Then closed to open complex transition. The initial binding forms a closed complex where DNA remains double stranded. Then the hollow enzyme locally melts about 12 to 14 base pairs around -10 regions, that means upstream of the transcription start site, to create open complex. It exposes the template strand inside the transcription bubble like this. So it opens or exposes the transcription start site to start the transcription process. Okay, and RNA polymerase works in the direction of 3' to 5', 3' to 5' direction. Okay, and it takes place on the template strand. Okay, next the phase 2 or step 2, which is elongation. So the remaining core enzyme moves along the template strand in a 3' to 5' direction. It synthesizes new RNA transcript in 5' to 3' direction at approximate rate of 40 nucleotides per second. Okay, so RNA transcription takes place in 3' to 5' direction, but new mRNA formed is from 5' to 3'. The enzyme uses antisense or template strand to build new mRNA, as I have already told you, meaning the resulting RNA sequence matches the coding or sense strand exactly, except the uracil part replaces thymine, because DNA have thymine and RNA have uracil instead of thymine. As the transcription bubble moves, DNA unwinds ahead of enzyme and rewinds behind it. Okay. Next, phase 3, termination. Termination or transcription stops when core enzyme encounters a specific sequence called terminator. Prokaryotes use two types of strategies to achieve this termination. So either prokaryotes follow row dependent termination pathway, which is otherwise called as intrinsic pathway, where the DNA template contains an inverted repeated sequence followed by a string of adenine residue, A stretch. When transcribed, the RNA folds into stable hairpin loop structure due to self-complementary base pairing. This hairpin physically stalls the moving RNA polymerase. The trailing RNA segment consists of weak uracil adenine bonds, allowing the stalled transcript to easily dissociate and break away from the DNA template. So it is one of two pathways for termination of transcription. Another method is row dependent termination. It requires specialized protein hexamer called row factor, which functions as ATP dependent helicase. It binds to a specific cytosine rich region on growing RNA called rot site or row utilization site. Row climbs off to the DNA strand towards the transcription bubble. When the RNA polymerase pauses at a downstream termination sequence, row catches up the complex and uses ATP or energy to unwind the RNA DNA hybrid, releasing the transcript. Okay. So what is upstream and what is downstream? The site near the promoter sequence where transcription starts is called the upstream of a gene or +1 site. And near the terminator, the site near the termination site is called the downstream of a gene. Okay. So with this, the whole steps or whole process of transcription is completed, including my lecture video here, and I hope this video will be beneficial for you all. And if you liked it, please hit the like button and share with your other friends, and please subscribe to my channel Sunanda's Tutorial to get more important videos on biology on this channel. Please subscribe to my channel Sunanda's Tutorial. Thank you.